Hi, this is Dr. Rosario, and this is lecture 9.3 on electron transport. This lecture continues our discussion of cellular respiration. So in this particular lecture, we're going to be focusing on the electron transport chain and oxidative phosphorylation. So the major goal for this lecture is for you to understand the role of electron transport in that overall process of taking the chemical potential energy stored in glucose and converting that energy into its usable form as ATP. So we're going to talk about electron carriers, oxygen, the proton gradient, and finally the ATP synthase that's going to produce our ATP. So in the last lecture, we talked about the different stages of cellular respiration. So I discussed glycolysis, pyruvate oxidation, and the citric acid cycle in detail for you. The fourth stage of cellular respiration is oxidative phosphorylation, and that's what we're going to focus on in this particular lecture. Now, oxidative phosphorylation produces the most ATP per glucose molecule. This process occurs inside the mitochondria. So we left off at the end of the last lecture with this chemical equation. The oxidation of glucose to carbon dioxide leaves us with 10 NADH and FADH2. Those are reduced electron acceptors, and a little bit of ATP. 2 ATP molecules from glycolysis and 2 ATP molecules from the Krebs cycle. Now, most of the energy that was stored in that glucose molecule remains on the electrons that were removed from it. So those reduced electron acceptors are carrying the energy from glucose. In oxidative phosphorylation, we use an electron transport chain to produce ATP. That electron transport chain then leads to a process called chemiosmosis. So let's look a little bit at the electron transport chain. So this is found in the inner mitochondrial membrane. It is a series of proteins that are embedded within that inner mitochondrial membrane. Remember that inner mitochondrial membrane has all these little folds called cristae in it, and those folds allow for a ton of these electron transport proteins to be packed inside a single mitochondrion. Now, these proteins are arranged in a sequence within the membrane so that an electron can be removed from an electron carrier like NADH or FADH and then passed from one protein to the next. Now remember, these electrons carry energy with them, and as they are passed from one molecule to the next, some of that potential energy is going to be captured by the cell. And I'm going to talk about how it's captured in a moment. Now, so as the electrons move through this electron transport chain, they are losing their energy. And then finally, the low energy electrons at the end of the electron transport chain, they are passed to oxygen, right? So that the final product here, you have 2 hydrogen ions plus half a molecule of oxygen that accepts 2 electrons and it forms water. So the electrons that we're using here, they came from glucose. Right? They're carried by our NADH and our FADH2. This is all occurring in the inner mitochondrial membrane. And we call oxygen the terminal electron acceptor because that is where the electrons finally end up at the very end of this process, right? So the electrons have moved from glucose to the electron carriers, to the electron transport chain, and then now they finally reach oxygen. But the question you should probably have now is, where did the energy go? If the electrons are losing their energy as they're being passed through the electron transport chain, where does that energy go? Well, it turns out that the energy goes to a hydrogen ion gradient. So as the electrons are being passed from one molecule to the next in the electron transport chain, hydrogen ions are being pumped from the matrix of the mitochondria out into that space between the inner membrane and the outer membrane. That's called the intermembrane space. And what we're doing here is we're creating a gradient. And a gradient is a form of potential energy. In this case, we have both a chemical gradient, the concentration of hydrogen is higher in the intermembrane space than in the matrix, but it's also an electrical gradient because we're moving those positive charges into that intermembrane space and out of the matrix. So here we've built up potential energy in this hydrogen ion gradient. From here, that energy is used to drive the synthesis of ATP. So there is another protein that we find embedded in the mitochondrial inner membrane. It's called the ATP synthase. And as the hydrogen ions move down that gradient, ATP synthase captures the energy and uses it to connect ADP and inorganic phosphate. We're synthesizing ATP. This is oxidative phosphorylation because it requires the use of an electron transport chain and the oxidation of our electron carriers. So the total yield of ATP that we get from aerobic respiration is approximately 30 to 32 ATP per glucose. 2 come from glycolysis, 2 come from the citric acid cycle, but the bulk of the ATP comes from oxidative phosphorylation. Now, it's really important to remember that oxidative phosphorylation can only occur if there is a terminal electron acceptor available. So in the case of our aerobic organisms, that means having oxygen. What do cells do if they don't have oxygen available? Well, that will be the topic of our next lecture.